Welcome to part 4 in the lactic acid and exercise physiology series, where we're going to discuss where lactic acid production affects exercise. This video will also cover the concept of lactate threshold. I would encourage you, like always, to cover the first 3 videos in this series and ensure you're comfortable with how ATP is produced in skeletal muscle. So far, I've only talked about lactic acid, and I've dropped the word lactate once in a while, but haven't fully explained it. Lactic acid is an acid, and like all acids, in the presence of water, it dissociates to its anion lactate and its hydrogen ion. So as soon as lactic acid is formed in the cytosol, it immediately dissociates, and therefore we cannot measure lactic acid. However, we can measure blood pH, which is an indication of hydrogen ion concentration, and blood lactate concentration. So why does lactic acid matter in exercise physiology? We know that as hydrogen ion concentration increases, the tissue and the blood surrounding the tissue becomes more acidic. That is, it has a lower pH, and this is going to have a number of effects in the cell. One of the things we know that a higher hydrogen concentration affects how myosin and actin bind to one another. And we know from numerous studies that this is actually going to slow down the cross-bridge cycle, meaning that muscle contraction is going to be inhibited. In addition, we know that more acidic conditions affect the enzymes in ATP producing pathways. They slow them down. And this is also going to inhibit cross-bridge cycling, as ATP is needed for a full cross-bridge cycle to occur. Finally, we've all experienced the pain associated with metabolites of ATP production, and it's suspected that hydrogen ions are one of the factors that may cause this pain. So the fact that we are unable to contract our muscles as quickly, that there's not as much ATP available, and that we're in a lot of pain is going to affect exercise performance. And this is why exercise physiologists focus a lot on lactic acid production. We are capable of measuring blood lactate, and this is going to be an indication of lactate production in the skeletal muscle, subtracting how much is being cleared by tissues such as type 1 muscle fibers, the liver, heart, kidneys, etc. In the previous video, we talked about several factors that are going to affect lactate production. One of them being how much oxygen is available in the mitochondria. The second, how much we're relying on glycolysis. And the third, how many type 2 muscle fibers are we recruiting. One factor we didn't go into detail is how lactate is being cleared from the blood. Tissues such as the liver, heart, kidneys, and type 1 muscle fibers contain the isoform that converts lactic acid back to pyruvate so it can be used in metabolism. If we have a greater predominance of type 1 muscle fibers, or if there's a greater amount of blood flow going to the liver, heart, and kidneys, we can better clear the lactate produced during exercise. So now that we've established that blood lactate concentration is determined by how much lactate is being produced by the skeletal muscle and how much is being cleared by other tissues, we can now look at the lactate threshold. And we'll define it as the exercise intensity at which there's an abrupt increase in blood lactate concentration. Here you can see on the y-axis, we've got the variable blood lactate, and it's measured in millimoles. On the x-axis, I've plotted exercise intensity, and here I've displayed it as percent of VO2 max. And you can see we've got resting, 25, 50, 75, and 100% of VO2 max. At rest, and in the early stages of exercise, there's a gradual increase in blood lactate as exercise intensity increases. At a particular point where blood lactate accumulates much faster, we call this the lactate threshold. So the point at which there's an abrupt increase in blood lactate concentration. Lactate threshold will occur at different intensities for different people. But what it indicates is that lactate production now exceeds lactate clearance by tissues that can use lactate. And this would indicate that there's either not sufficient oxygen, glycolysis going too quickly, there's a greater recruitment of type 2 muscle fibers, or there's a lack of blood flow going to the clearance tissues. And what it does reflect is the ability to sustain oxidative metabolism, being that the ATP demand is so high that it can no longer be obtained through aerobic ATP production only, that glycolysis is sped up, and that phosphocreatine is required. As I indicated earlier, this lactate threshold will occur at different intensities for different people. So for example, lactate threshold may occur at 80% of VO2 max for a trained individual, where it may occur around 50% of VO2 max for an untrained individual. And this is going to reflect that a trained person can better use aerobic ATP production to sustain exercise. This video will not cover how oxygen in the mitochondria, reliance on glycolysis, and recruitment of type 2 muscle fibers, as well as clearance, is affected by endurance training. This is a whole other series in itself. To recap, in the cell, lactic acid dissociates to form lactate and hydrogen. Blood lactate can be measured, and this will indicate the balance between lactate production and lactate clearance. Why lactic acid matters to exercise physiologists is because an accumulation can lead to deficits in performance by affecting muscle contraction, ATP production, as well as causing pain to athletes. Endurance training can influence the production and clearance of lactic acid. I hope you found these videos in the lactic acid series a good start to your understanding of lactic acid and exercise. For those advanced students of exercise physiology, you'll want to delve into some of the research